efficient use of population genomes.
Okay. So I'm going to talk about the use of all this new DNA sequencing data for studying the variation in our genome sequences, genetic variation. And Leo mentioned yesterday that there's new technology for DNA sequencing, which is transforming our ability to collect DNA sequence. And I think Matt this morning talked a little bit about how we're using new technology sequence for finding copy number variation. But I wanted to put it into context to start with. So DNA sequencing has really driven genome science, certainly, and the whole, and to some extent, I think, biology, arguably, for the last 20 years. And really, there's been an exponential growth in our total amount of DNA sequence. So here we have a plot showing the amount of publicly available DNA sequence in the public databases, starting over 20 years ago in 1984, when there was 1 million bases, which was a lot in those days, to, in fact, it finishes around several years ago, 2004. And you can see that this is on a log scale, log number of base pairs. There's about a 16-month doubling time overall in that period. So this is one of these technologies rather like computers, where we see exponential growth in the technology. And there's been a case I heard made, in fact, that this is possible because DNA basically is information. The physical realization, of course, in the cell is as nucleotides, chemicals. We represent it as in computers digitally, and but physically one needs extremely small resources to represent DNA. We're actually a long way from our limit, the limit of being able to capture DNA sequence. So there's a 16-month doubling time, but actually you can sort of see this as two phases. There was a first phase up to here, which is remarkably linear on the log scale, and this was using sequencing by hand. So that's exponential growth in a sort of hand-driven process. And at that point, around that point, the first generation of sequencing machines arrived, and they produced a change in the slope. And then there's a little blip up here, and that blip is the Human Genome Project. That's sequencing the human genome the first time, which was an enormous effort, huge concentration of money and minds and effort internationally. And it just made a tiny little blip on this exponential growth curve. And I think the second half of this is more like doubling time of once a year in DNA growth. But as we've heard already, I think, at this meeting, that there's new technology, and my prediction would be that this curve is going to accelerate again. And that's good for science, good for biology. There are many things that we can do with a sequence. I think there are a lot of issues to do with handling data, noisy data, in vast amounts, which is essentially the subject of this meeting, which are going to come in the next few years. And all things close to the genome, which can be represented in nucleic acids and DNA, will be able to be assayed in this sort of way. Okay. So this is a slide, repeating what you saw yesterday a bit. Essentially, we used to obtain the sort of what's called old technology. I think maybe I'll get the mathematician's version of laser pointer. Much more reliable. We got about 80,000 bases in a run, cost about $1,000 a megabase. These are all the new technologies, first introduced in 2005. We got 1,000 times more data per run, a tenth of the cost. And the next sort of generation coming just 1 year later was another factor of 10 in amount of data per run, and factor of 10 reduction in cost. So there's a 100-fold reduction in cost that's taken place over a period of a few years. And there's a second machine that was introduced late last year with very comparable performance to the Illumina Selector. These two are sort of fighting it out just now. There are other companies trying to get in on this game. And the expectation is that these will drive down this cost. We saw a value of $5 a megabase yesterday, and the hope is to drive this down by another factor of 10 to 100, which will give us the $1,000 human genome for $1,000. So just to give people some idea of how all these new instruments work, they all look down, imaged through a microscope, a surface which has got a sort of two-dimensional array, either random array like this or a less random array of effectively points. These are little clusters or beads or something punctate, more or less, each of which represents a starting molecule. And the idea is we're going to read the sequence off by looking at one base at a time, sequentially. So here what I'm going to do is blow up a section of this and show you a series of, so we're using here a 4-color system. This is the Illumina system. It's four colors, so what's happening is you're using 4 different dyes, fluorescent dyes, each of which will tag essentially one of the bases. So we're looking at a single base here, let's say the first base in the sequence. And the 4 different colors mean that some are A, C, G, and T. I don't actually know which color is which base, but doesn't matter that much. So here we blow up, and here we can go along. That's green, green, blue, red, green, blue, yellow, blue, yellow, green. And it's effectively exactly that way you can read off the DNA sequence of that particular molecule, and you can do the same in parallel for all the other molecules on the slide. And there are different approaches to scanning and so on. Those things that took about a second here might take a couple of hours or an hour. Depends on the technology. But all these new systems work in this way. And the extra dimension, essentially, of looking down on a two-dimensional surface and using time is what's giving us the ability to obtain millions of sequences in parallel. OK. So what can we do with this data? Well, as we heard yesterday, I mean, in fact, there are many things that we can do with this data. But particularly for genome sequencing, we can ask what we can do in that context. Well, from my point of view, people sort of say, oh, well, genome sequencing is all done. But from my point of view, we've just done phase 1 properly. We know how to obtain a reference sequence. This is the last 10 years or so. We got first a bacterium, then a yeast, then a small animal and human genome. But that just gives us a reference sequence for an organism. Effectively, it's a sample of 1 from the distribution of sequences. Each of us has, in fact, 2 copies of the human genome, 1 from our mother and 1 from our father, and all those versions are somewhat different from each other. And the reference sequence is just a sort of archetype. It's a single version. So phase 2 is to characterize variation. And there have been a number of projects in that direction. There were projects to find SNPs, find single points. SNPs are single nucleotide polymorphisms. They're the single positions at which there is variation. And typically, between any 2 copies, there's a difference about every 1,000 bases. Now, those differences are in different places between different pairs. So there's more than 1 variant per 1,000 bases overall. And we now have got about 10 million of these single nucleotide differences. There's a second sort of phase to look at covariation. If you have a variant at this position, is the value of that variant independent from the value at another position? And the answer is it's not. They're correlated, and the correlation is due to ancestral sharing of genetic material. And that's called linkage disequilibrium. And the HapMap project, which finished a couple of years ago, 2 or 3 years ago, began to look at this covariation. But that was only over a sample of a couple of hundred people, like 60 people essentially from 60 Europeans, 60 Africans, and 90 Asians. So it's a relatively small sample, and also it looked at only a fraction of these variants, about 3 million in the end. And then, as you heard this morning, there are multiple efforts to find copy number variation. This is variation not at single positions, but which affects larger regions of the genome. But that's very much ongoing and incomplete, as I presume you heard. So all of this is incomplete, and I would say the ultimate goal is the description of the distribution of complete sequences in the population. And it's that would, if we had a better picture of that and description of that, that would underpin all our work in human genetics. So, the solution to that is to get the sequences. And the reason we haven't done it previously is that it would be too costly with the old technology, and with the new technology, it's now possible to collect data. And so we can just go physically and sequence lots of people to sample more deeply the sequence. But we want to do that efficiently and sensibly. And essentially, this is a question of how do you describe a distribution effectively. And there are also going to be issues about how we use the information for the downstream human genetics. So as an example, though, which is going to sort of motivate things, and in fact is pretty much the target which we're trying to get to, at the moment, we'd like to find all the variants and their pairwise correlations down to a 1% allele frequency. So this matters. I now realize I should have included another slide, but because some variants are present in many people. There may be one variant where half of the genomes in the room have a G and half have an A. And there's some positions at which there's a variant which has 10% frequency, so 10% of the genomes have a G and 90% have an A. And there's some at 1%, and there's some which are private. There's some where you might be the only person in the world, possibly, who has a G, and everyone else has an A. Actually, there's enough people in the world that almost every possible variant that isn't lethal is present in somebody, but it's around that level. Okay, so that's a good, the 1% level. Realistically, actually, we now have things at about the 10% level. Getting down to the 1% level would be a good goal, and it's actually a classical goal. People talk about polymorphism as variation at the 1% level. That's not mathematical polymorphism, it's genetic polymorphism. Okay, so I'm going to talk about a pilot project to use the new sort of data for human genome resequencing, which raises some of the issues involved in resequencing using the new technologies. Then a second project where we looked at how to describe a population variation, not in humans initially, because that's an expensive and large-scale thing, but in yeast, which has got a genome 1,000 times smaller, or no, 300 times smaller. And a sort of model-based analysis of that, and then a proposal to scale up to 1,000 humans. And there are design questions there which are very alive in the community, and which I think are a kind of good subject for this meeting in a sense. Because there are statistical issues in how to most effectively get the data. So how completely and accurately can we find variants with the new technology? Well, firstly, true novel variants are going to occur approximately every 5 to 10 kilobases. I said that variants occur once every kilobase or a few hundred bases, but we found quite a lot of the common ones. So in a particular sample, that's about the rate. And that actually sets quite a high standard. We can't afford very many false positives. If we had a false positive every 10 kilobases, which actually was the original goal standard of quality for the human genome, then we would, 1 in 2 detected variants would be an error. So that's a useful thing to bear in mind. We probably want at least a low false discovery rate about at that level. The second thing that's important from a sampling point of view is that all these methods are what we call shotgun methods. Basically, they sequence a random sample of single molecules. So the way we sequence the human genome is that we take DNA from the entire genome and chop it up into random pieces, and we pull out random pieces and sequence them. And in fact, it's sampling with replacement, essentially, from the human genome. And that means if you sequence 3 gigabases, you won't get 1 copy of each of the 3 gigabases. You get essentially, it's a Poisson-type process. You get Poisson sampling of depth at any locus. And that's at best. If there's any bias, so that some areas are more sequenceable than others for some reason, then we'll get even worse dispersion than Poisson. And then we have a problem that every human has got 2 copies, and if you just take all the DNA from a human, and even if you sequence 10 copies at this position, or you're going to sample, each of those 10 will be a random sample from the 2 different versions or haplotypes. So you need to have quite a lot of depth in order to guarantee that you've seen both haplotypes. So if you want to say what versions a particular person has, it's not good enough just to cover it once or twice, because you can't be sure that you've seen both copies. And that's unlike previous methods of sequencing, which basically what they do is they essentially start from a very large number of molecules, and so you're getting pretty much a proportional representation of both versions. Okay. So anyway, these are sort of fundamental limitations. How are we going to analyze this sort of data? Well, we're going to take the initial data, and we're going to get a little bit of sequence. And of course, being an experimental system, we don't actually get the correct sequence of what we started with. There are errors on that. But we can get some sort of confidence values, which we call base qualities. So at each position, we have a measure which typically, and I'm going to talk about these a bit, so we use a sort of metric. It's minus 10 log 10 of the probability of an error. So a Q value of 10 would mean a probability of error of 0.1. A Q value of 20 would be 1 in 100. Q30 is 1 in 1,000 chance of an error. Q50 is 1 in 100,000. We kind of want to get to Q50. But the raw data is not going to be that good. Typically, the raw data is Q30, where it's good, and Q10, where it's bad. So we're going to get these reads. We then have to map them to the reference genome. They're going to be little pieces of sequence. We don't know where they come from. And there are a lot of issues in that. Leo mentioned them, computational issues, but there are also statistical issues, given that there are errors. And then once we've decided that all these reads come from this place, we want to look and decide actually what the underlying, infer what the original starting sequence was. And that's called calling the genotypes. So what are the sources of error? Well, we get sampling error, as I've said. We need high average coverage to guarantee enough read depth to see both alleles sufficiently often. Bit of a mouthful, but so we need depth 9, for example, to ensure that the probability that both alleles are seen at least twice is bigger than 95%. We're going to want to see both alleles at least twice because we probably aren't going to believe something if we only see it once, because it could be an error. And so somewhere around depth 10 to have high confidence of seeing both alleles twice. Now, that's a depth at a particular position. What sort of average coverage do we need in order to ensure that we get a depth of 10 almost everywhere? We need an average coverage of somewhere in the 20s to have that kind of confidence. So even from a sort of simple sampling analysis, it's likely that to get high confidence in the sequence of an individual, we're going to need something in depth in the 20s. Then there are sequencing errors. Sequencing errors arise from sample amplification, from chemistry. That means we start with DNA from the person, but we don't actually sequence that. We sequence something that's made out of that, and it could be errors in the copying process. There can be errors in the chemistry used for sequencing. There can be errors in the measurements on the instrument, the light, the image analysis, and in the software which does all that. And unfortunately, the errors are also not independent. So if at a particular position, given that there is one read has produced an error, it's possible, it's more likely that another read has produced an error. Now, there's interesting questions about this whole business of calibrating these quality scores in this context, and I could come back to that, maybe in discussion afterwards with people. One could specify that one of the goals of quality calibration is to give quality scores that are conditionally independent. So that, and then third, there are problems with mapping and assembly. So as I mentioned earlier. Okay, so sort of having got that context, let's see, what we can do and And we've worked on a project with Illumina, with the company, in collaboration, just using a flow-sorted X chromosome. So this is one chromosome, one of the human chromosomes. It's 150 megabases, so it's about a 20th of the human genome. And you can flow sort because physically the chromosomes are different sizes and they're different weights. They fly through the air in a flow sorting device differently. I guess actually it's it's not the weight, it's the amount of fluorescence that they have, the amount of DNA they contain that you can separate on. So you can separate out a chromosome, and this comes from a single European female. And so there's 2 copies of the genome. And we collected 3 different data sets, and I'm going to talk mostly about the last of them, which is about 45-fold depth on average. And this has been a sort of driver for our analysis. So we developed a program MAC, which maps and calls the variants. And this is really the work of Li Hung, who's a postdoc in my group. And the approach here, we heard Leo mentioned yesterday that Eland, and in fact most other programs that people use, will only map a read if it has a unique best match. So if there's several places that it could match which look equally good, then they'll say, I don't know where it goes. Our approach is a bit different. We always place it, but we assign a mapping quality. So that's the confidence that we have under a model, sort of sampling model with errors, that it maps in this position, or conversely. So Q20 mapping quality would mean that we think there's a 1% chance that we've made an error in placing it. Very analogous to the base quality. So we use those mapping qualities in combination with the base qualities when we score the genotypes. So it's another measure of how much we can trust this data. And we map paired reads jointly. You can get 2 reads at once which are nearby each other. That's called a read pair. I think you heard about those this morning. And when we do that, about 7 million, 20% of the reads map differently as a pair than singly. So, one point that we think is important is that mapping reads in pairs, the reads are quite short. They're only 35 bases, and so there's quite a lot of uncertainty in where to place them. 4% map differently as pairs with more mismatches. That means that if you place them in the place that was best, that would have been incorrect. Actually, the place that's more correct according to the pair is one where there are more mismatches than the other one. Now, most of these actually are in places where most of the mapping is difficult. Mostly the mapping is difficult because there's a lot of repetitive sequence in the human genome. There are quite a lot of the genome, about 40%, is composed of sequence which looks like some other piece of the genome, more or less. But 0.05%, that's 1 in 2,000, which may seem not very much, but it's quite a lot in terms of this sort of error rates that we want to obtain. Mapped, based on a single N, we had a high quality, Q30 or better, confidence in placing it. And so these essentially were all put in a best unique position. And once we looked at the read pairs, we moved them to a worse position. So these are ones which I think Eland would have treated as confident, but which shouldn't have been confident. Of course, it's not so surprising that 1 in 2,000 of the things mapped with an error rate of 1 in 1,000 are misplaced. But I think it shows you're not as confident. Finding a unique best match is not confident enough, in fact, for calling things. Okay, and then we have a Bayesian algorithm that models the error dependency for calling the genotype. Let me go into. So this is just an advertisement for extra features of MAC. So MAC is available from SourceForge as a public source, and there's a manuscript submitted. You can simulate in order to generate data sets for testing with realistic errors that come from real data. There's the second sort of machine, the AB solid machine, generates data in some funny transformed space, essentially. And for those who know, they'll know, and those who don't need to know. But MAC can handle that sort of data as well. There's a sort of sequencing that allows you to explore methylation, which we heard about yesterday, and Mac has got a version that handles that type of data. And there's a whole lot of kind of visualization and other support tools around the basic resource. Okay, so what happens when we do all this? Well, firstly, I said that you'd expect the depth distribution to be Poisson, and it's of course not as good as that. So here's in gray the distribution of all the data, the depth. So this is looking across the X chromosome, how many reads cross each point. The mean is about 45. The gray is everything, and this line is Poisson. Actually, the gray is a bit more. They're not normalized exactly, or these two are normalized, I guess. So anyway, you can see that the gray is over-dispersed. There's significant, upsettingly, there's more, which is more of the genome has got low coverage than you would expect, according to Poisson distribution. Now, part of that is due to uniqueness. But if you remove all the mapping issues, if you take the things with high mapping confidence, essentially which map uniquely, then you get this curve, and it's still over-dispersed. So part of that is due to GC bias. Ewan said that everything in the human genome has got a variation with GC composition. And that's true for the biological things, but it's also true for the experimental data. And what you'd like to see, so this is plotting according to GC composition. This is from 10% up to 0 up to 100, but it's in percentile units. So the average GC composition of the human genome is about in the high 30s or 40%. The median is 36 or something. And what we can see is that at high GC and at low GC, a bit less so, this particular library has lower representation. It doesn't represent high GC DNA so well. This line here isn't flat. Actually, this library isn't so bad. Some libraries are very bad, and they kind of skew down here, and they come down here, and that's a property of the sample preparation, and so on. So we can look at the sort of variance. If this was a true Poisson, the variance over the mean would be 1. For the whole chromosome, we get a variance over the mean of about 6. Unique sequence 2.2, and after GC correction, the sort of residual variance over the mean after compensating is about 1.6. About half of the variance here is accounted for. Excess variance is accounted for by GC correction. And we can actually, if we want to, we can model that excess variance with a gamma mixture distribution. And that can be useful for if we're wanting to sort of model the sampling for, say, looking at depth variation to study copy number variation, as Matt was talking about. But it doesn't help us in the sense of sampling. You can't generate more data in the regions which have got low depth. Okay. MAC alignment and SNP calling. About 280 million purity filtered or reasonably good reads. 96% of them mapped to the human genome. 97% of those were in read pairs, which is 90% mapped to the X chromosome. That's about what we'd expect. About 10%. The flow sorting isn't perfect, and you get some bits of other chromosomes. And there's a technical issue that we get some duplicates from the way that the library was made. So we end up with about 200 million good reads. And we called 106,000 SNPs. And we called them on about 93. So we can sequence about 93% of the genome. The other 7% is about 10 or 11 megabases, is not accessible because it's too repetitive. It's not unique enough that we can't get any kind of good handle on it. And about 85% of those were known already. and that number looks a bit high, but actually we know that this sample was used for finding variants earlier. So if we started with a real brand new person, that number would be smaller. There's more novel material than that. And it's about 1 every 2 kb. Differences, and I told you that people were different 1 every 1 kb, but that's on most of the genome, and the X chromosome is the sex chromosome, and the heterozygosity rate is lower because of population genetic reasons, and that's about what we expect. And we also called a lot of small indels, about 10% of the number of point mutations. These are short insertions and deletions, and that's also what we'd expect. Though our methods for doing that are probably not as good as for point mutations. So how accurate are we? There's another way of looking at variation at known positions. You can sort of query known positions which are known to be variable and ask what the genotype is. And so there's a chip, 550,000 SNPs genome-wide, 13,600 of those on the X chromosome. If we get that data and compare with our calls at those positions, 12 of them we filtered out because we weren't able to call them. We didn't, and we only made a single error. And if we go into a mode, and that's where we were in a mode that was sort of de novo sequencing. If we say we know that there's a variant here, we're going to ask what the most likely value is of that variant, which is an easier task because you can set a prior, which is a high prior for there being variation. Then we actually make no errors. So that's very good. I mean, that's kind of remarkable, actually. It's even remarkable that these two entirely different methods agree so well. But this is at 45-fold depth. What happens if we can subsample now and ask how things go if you have lower depth? So here there's a sort of plot of this, and at, say, 20-fold depth, first you mask out a couple of hundred or 100, which is 1.5% or so, and then you're starting to make more errors as well, maybe 0.1% rather than 0.01%, so 10-fold more errors. And if we go down to 10-fold coverage, then we mask out 10% that we can't see, and we're making maybe a couple of percent errors. So what we're seeing here in a way reflects what we said right at the beginning, that you need very large depth sampling to get high confidence results. One good thing is that all the errors, even down at this level from MAC, are undercalls of heterozygotes. So these are where the person has got two different values, and we call it either as all A, they're an AG, or all G. So they are what you'd expect from a method that's sort of being driven by the sampling properties rather than by errors in the data. And if we look at the published version of the human genome that's come from Craig Venter, that was done with old-fashioned capillary sequencing, that was only 8-fold deep, and they got, with pretty much the same assay, 91% genotype match. They made 9% errors, which was a lot worse than this. And frankly, that wouldn't really be tolerable in terms of genotyping by most geneticists. Although it's a triumph, I should say, to achieve that. And you've heard already about obtaining structural variation. That basically from this type of data. So this is, I think, Matt has been involved in this analysis, and Claudia, who's at the meeting and has a poster about this. You can use this sequence data in two ways, really. One is read depth, places where there's lots of extra copies. And the other is with read pairs, where there's a part of the genome that isn't represented, and there's all these little green bars are sort of happy read pairs where the reads are 150 bases apart. And here are a lot of cases where the reads map, I don't know, a couple of kilobases apart, and that's because that couple of kilobases is missing from the sample. So, okay, that's all very well. What are we going to do? We've learned quite a lot about how we can sequence individuals. But we need to sequence lots of people to find almost all the 1% variance. And sequencing each one to high coverage, say 30x, which is about where what we think we'd have to do to get high accuracy sequence, is expensive, even with new technologies. So is this the best way to describe the distribution of variation in the population? Is there a more efficient way to discover variation? Well, what we're interested in shared variance if we want to describe the distribution of the population. We probably aren't interested, in the first instance, in individual variance that are private to an individual. So we can combine data across individuals, and arguably we should. And an alternative approach to sequencing each person to high depth is to sequence more people, but at low coverage. We can use the same amount of sequence. And sequence is still expensive. We're talking about tens of millions of dollars worth for this kind of project, which is in the scale of what people have been doing and is kind of justifiable. But you don't want to be wasteful, or grossly wasteful. So the principal idea is that it's more efficient to share data across individuals, even if you are not so accurate on each one person. So it's all very well having that as a kind of arm-waving idea. Can we show that it works? Well, we can both do that by simulation, in theory, and also we can do pilot studies in practice. So we did a pilot study in yeast, small genome. We can get haploid clones, so we get single versions per strain. There's none of this problem of diploid, 2 copies. There's a whole strong yeast genome community who will use all the data. It's important in its own right. And we sort of gave ourselves a budget of 100-fold total coverage, and we did some strains to 1x and some to 3x, and we actually sequenced 2 different closely related species for evolutionary and population genetic reasons in terms of the design. And we started this even before we had the new technology, so most of the data was from using ABI. So if you sequence the low coverage, of course, you don't cover everything. So here's an example of a little piece of the genome. Here are places where there was no data. These 2 strains had no data at all in this particular part of the genome. And here we've got a read that starts. There's missing data here. And at each position we have a base call and a quality value. There's a number between each of these below. And all the things highlighted in color are differences, and but some of them might be errors. So can we identify which of these differences are real, and can we also recover the missing sequence? Can we infer the missing sequence? Now, what we're going to do is combine data across strains. So we're going to look at the column down here. And if we knew that the way that all the strains were related, this would be relatively easy. So here we have a case where we have 7 strains overall. There's missing data here. We see some A's and some T's. Now, if you didn't know about the history, so there were 3 A's and 3 T's, you wouldn't be able to guess what the value here was. Also, this T is a lower quality. It's got less confidence to it. So if you look at, if they're related that way with a tree, then you could be quite confident, essentially, that it would be most likely that there'd been a single mutation here that had given rise to both of those T's. So that was likely to be an A. And this could have been another mutation to give a T, but because it was unconfident to start with, it's actually probably more likely that it's an A as well. Okay. So all very well. If we had the tree, life would be easy. Unfortunately, there's no single tree. The tree varies as you move along the genome because of recombination between strains. So trees that are nearby on the genome are related to each other, and it's that gives rise to this correlation structure, this linkage disequilibrium. So we can try and identify possible recombination points and segments between them that share ancestry, and so we can actually try and reconstruct what the tree is. And I'm running short of time. So there's a structure called the ancestral recombination graph that describes the way that populations are related. It describes this sequence of trees as you move along the genome. And here's an example with 4 sequences. They're the same at all the dots, and they're different, these positions. So there's 3 positions where they differ. You can express it in sort of binary form. And here's a possible tree that explains the first position. So A is a C, or 0, and all the other 3 strains have got a mutation that takes place, gives rise to this one, so there'll be that mutation. And the same tree can explain the second position here. C and D share the mutation. But there's no way to put a single mutation on that tree which will explain the third position, where B and D share a mutation. You need a different tree, for example this one, where B and D share a mutation and A and C don't. So how do you, sort of flip from this tree to that tree? Well, that can happen. It can happen biologically. What happens is to the left of this point, that tree applies, and to the right, this tree applies. And that's due to the recombination event that has brought together material from 2 different ancestors. We can draw it like this. So we can draw a graph that sort of bifurcates going down and going up. There's a sort of symmetry to it. And that graph's called the ancestral recombination graph, and it describes where all the DNA comes from in these samples. And in general, an ancestral recombination graph is the complete description of the history of the set of sequences. It gives a tree at each position. And so it kind of captures everything you would want to capture for doing genetics. Unfortunately, there are lots of possible ARGs that explain the same data. And so it's an ill-posed problem to find the correct one given a data set. But some are more likely than others. So there are sampling approaches, but correct sampling is hard. So we have a heuristic algorithm, Margarita, which is iterative and greedy, although it has some stochastic elements. And it basically puts together a plausible, reasonably likely ARG given data. And we've used it for human disease mapping. And in yeast, the data is such that it should work fairly well, as in fact as in humans. And once we've done all that and got a tree, we can then use standard probabilistic inference methods on trees to infer missing values and correct values. So basically we have a tree here with a mutation rate on it, and then we have an observation process with a measured error rate which we can use to fill in the missing values. So here's an actual example from yeast. These are the observed values. There's G's over there and C's over here. This is the tree that we infer with branch links. Basically, you'd expect there to be C's down here and G's down here. And we can infer missing values like this. So here's the example I showed you earlier. What we've done is we've filled in missing data. We've increased the confidence at most positions, and we've corrected errors. So that was an error. This is a real call, that's a real call, and that's a real call, and we can call all the missing values. And we can use those to look at the correlation in LD structure. What does this do to the error rates? Well, it's quite easy to measure the error rates for this process. So the error rate where data were present dropped from 0.84% down to 0.12%. So we improved the confidence. The error rate on fill-in is about 0.36%. That's better than the error rate where we had data before. And in fact, this is an underestimate. This is the way that we measured the errors, and it's also on a small number of strains. So we've also extended this to simulated data sets. For example, for human, we've taken 100 people, 1 million bases, sampled at 1x coverage with errors. We reconstruct that. We find all the SNPs with minor allele frequency bigger than 5%, and 95% of the SNPs with 94% of the SNPs with minor allele frequency bigger than 1%. But very importantly, we're not good at finding the really rare things. We can only find the things which are shared. So once you get down to below 1%, there's a sort of our detection rate drops compared to high accuracy sequencing on single individuals. Our fill-in missing, it's always true with simulated data, things look better than they do with real data. So 0.01% error on filling in, and below that, 0.006% on correcting data. And this type of reconstruction, which is based on sort of ancestral shared ancestry, is better than more simplistic approaches that would just say, trust everything that you've seen twice, which would tend to overcall and be less confident. So before I move on, I think we have a method which can use those sorts of things. There are other methods that also use the shared genetic structure. This is a picture from a paper by Sheaton and Stevens. There's a variety of methods. So what happens with these ancestral structures is that you tend to have a sort of mosaic structure to the genome. These are a set of individuals, and the different colors indicate that in this region, all the ones that are pink share ancestry, or share pretty much the same sequence, and the ones that are green, and then as you move along, you get points where things flip over. And you can model any new individual as a sort of mosaic combination of pieces of other individuals. And there's a set of methods by Matthew Stevens and Jonathan Marchini and Gonzalo Becasis and others that do that. And they behave very similarly in terms of filling in missing data and imputation. Sort of they're probabilistic models of the relationship between sequences. Although it's not clear how they'll behave actually as you go to lower allele frequencies, I'd say. So given that kind of background, what are we actually planning to do? Well, earlier this year we established the 1,000 Genomes Project. It's an international consortium to generate a high-resolution catalog of human variation. There's 5 sequencing centers at the moment in America, UK, and China. There's lots of people involved in the analysis from all over the world, including some of these centers. There's a big sampling and ethics kind of effort to ensure that we're doing this in an ethical way, and we don't, because we've got data on specific people, that's done with proper consent and so on. And of course there's funding from NHGRI, part of NIH, and Wellcome Trust, and the various Chinese government and local government centers. And the aim is to produce a public open resource like the Human Genome Project or HapMap. Anonymized samples, and there will be cell lines available for further experimentation on the same individuals. So the primary goals are to discover shared variation and characterize it by allele frequency. We want to essentially find all variation down to 1%. That's the target. Or say 95% power down to that level. To find structural variants, and in gene regions to go even deeper, to get to even a lower level of allele frequency. And we want to place the variants in their haplotype context. Okay. How much more is there to find? About 15% of the SNPs found are novel, typically biased towards low frequency. So if you look, here's a study where we sequenced 48 people. About 2/3 of all the variants which are below 10% frequency were novel. About 20% of the variants in this region, and 10% of the variants above 0.2 frequency. So we found most of the variants that are above 0.2 frequency, but only a minority of the ones below 0.1 frequency. And our knowledge of the ancestral correlation structures is definitely incomplete, and same for structural variation. So there's quite a lot more to find. As well as just finding this variation and characterizing it for human genetic studies, there are some additional goals. So one is just to understand human history better, regions under selection, the history of the human genome, study of processes of mutation and recombination, and population differentiation in history. And importantly, we also want to improve the human reference sequence, which isn't perfect at all. We want to find and fix errors. Also, the human reference sequence isn't complete. Any one person has bits of DNA that aren't present in the reference. And we're going to find quite a lot of that, and we'll have to put it in and somehow represent that. And there's a whole consortium aiming to do this, and in some sense the two projects are sort of merging or tightly related, so that's an important goal. So how are we going to go about it? We're going to pilot this approach of using low coverage samples. So we're going to take 3 sets of 60 people at 2-fold coverage, one from Europe, one from Africa, and one from East Asia. It's 180 people. We're then going to do some people to high depth so that we can have confidence. It says here 20x, but actually we're going to end up, at least for some of these, the children in the trios, going up to about 40x. So we should get very accurate sequence. Doing trios, that's with 2 parents and a child. We can also look at the inheritance patterns and some other things. So there's some added benefit to doing things this way around. And then the pilot 3 is to pilot this in-depth approach to genes. We study 1,000 genes in 1,000 people. And we're going to use different technical approaches, different machines and protocols, and develop and evaluate methods for data collection and analysis. The idea is that this will all happen this year, and then based as that's coming out, we'll design the main project, which should start at the end of this year or beginning of next, which will involve additional samples so that we can. So, in fact, that's about where we are. We're just beginning to get data. We're collecting data. We started probably on the order of 10 people. We've collected data so far. We're about to, in a month's time, we're going to meet and discuss what we've learned from that and how we're going to go about what we need in detail for the main study design. And there are some open issues. So our reconstruction from low coverage data, it looks good, but it's model based. And a key question is it supported by the pilot data on humans? And in particular, what's the right trade-off for coverage versus number of samples in that discussion that I gave? So I was looking at before at about 1-fold coverage, but is it better to go 1-fold coverage on 1,000 or 2-fold on 500 or whatever? And a sort of separate but related issue is, even if this works just for detection of new variants, will the lack of high confidence data on each individual create problems? So let's say there's something new that we don't know about going on, or we haven't properly characterized. So it's not part of our model. If we have a model-based approach that studies uses weak data, we might miss seeing something that deviates from the model, essentially. And secondly, population genetic analysis, which is part of the aim of the project, classically it's quite dependent on high-quality data. It's very highly dependent on singletons. So the most basic thing that people want to measure is what's the population mutation rate, theta. And the estimators for theta are dominated by the singletons, the things that are seen once. But our method of discovery of variance relies on them not being seen once, but being seen many times. We actually don't see the things. We don't measure the things that are only present once in our sample. So in a way, we need, and I think the field needs, new population genetic estimators which are robust to low-quality data. And it may seem that this is kind of driven backwards by this project, but I think that's true in general. We're going to be able to produce vast amounts of data with the new technologies. And you might think, well, that'll mean that we can completely oversample everything and get very high quality. But I actually think it's going to be the other way around. We're going to want to have data from very many samples and somehow combine that data, but it won't be definitive on each particular sample. So in general, I think the field is going to want to sort of work with a stronger model of error in its primary data, probably with a likelihood-based estimators. And in fact, so I think in the community there's some of these ideas floating around, and that's quite open at the moment. Okay, so I've just about run out of time. I wanted to raise those as issues. So the conclusions are: we need high depth for high accuracy on one individual, but we can get high accuracy. It's a good message actually from the first part of the talk. We really can be get very highly accurate information. On the other hand, if we want to be efficient, we can, in terms of distribution, fitting the distribution, we can build a picture of population variation with low depth on many individuals, and that'll probably be good down to some level, 1% in the case that I I'm kind of aiming for, but it's probably a question of design how to get at this. But it's going to be poor outside that. There's going to be a trade-off, poor for private or rare variants. The 1,000 Genomes Project will construct a human genetic variation reference genome. We're at the point now where we're discussing the final design based on the pilot project. We've got a pilot project underway, but in the next 6 months or so, I think we're going to want to finalize the design for the main project. And I think in a way to support all these, we've got these methods, this ARG-based method that I discussed that's heuristic somewhat, these mosaic copying processes, which are probabilistic models of genetic variation in populations, but which I think are biased to certain common alleles, common variants. And I think, although there's been work in this area in the last 10 years or so, or 5 years, I think that there's going to be progress. There's quite a lot to be done in getting better statistical models of sequence variation, and they have to be computationally tractable. They have to be things that can be run on 3 billion bases, on 1,000 samples, on very large amounts of primary data. So that it's not good enough. Actually, the statistical genetics field had a history before data was collectible of having models which you ran MCMC for 2 weeks to deal with 30 samples and on 30 positions. And now that we have data, we have to worry about computational efficiency. So like computer vision or something. Okay, so to finish, there are lots of other people involved in all this. These are people sort of who've been directly involved in my group or Claudia's jointly with Matt. These are other people at the Sanger Institute who, and I think I'll pick out Li Heng as having developed the MAC software and the mapping and base calling in particular, and Mark having developed the ARG-based tree analysis. These people have all been involved in data collection, or Nigel Carter and B in sample preparation and advice. These are the collaborators at Illumina. David Bentley and Mark Ross are the kind of principal investigators, and Kira and Tony in the data analysis. And these people are all involved in the yeast project. Ed Louie is the kind of co-leader at Nottingham, and groups also at Imperial. And then the 1,000 Genomes Project is one of these international consortia with 1,000 people, it feels like. Not that it's actually, there's an awful lot of people all separated out into different groups, given that we haven't done very much yet, but I think there's going to be plenty to do over the next couple of years. So thanks very much.
A sudden thought came to me. The ability, people like Svante have really shown that you can now sequence ancient DNA, and does that change is there any, have you ever thought about deliberately going back in time with sampling? I mean, to sample back in time, has that ever crossed anybody's mind as helping this process or not? Probably doesn't, I guess.
Right, so what's going to happen is if you sample people now, if you sample 1,000 people, at any one position, there are 1,000 different versions of the genome, but those coalesce quite rapidly, so that if you go back 10,000 years, you probably only have a few hundred people. If you go back 100,000 years, you probably only have a few people, like 10. Now, if you took a person from 100,000 years ago, that would be 10. There would have been, 100,000 people around, or 10,000 people around at that time, 100,000 years ago. But there's only 10 of them that contributed to modern genetic diversity. Now, if I sample somebody else from 100,000 years ago, the chances that they're one of the ones that was an ancestor of people alive today is slim. So it would be nice to sequence an ancestor,
but it doesn't.
But the odds are that you wouldn't sequence an ancestor. You'd sequence somebody on a branch that became dead. And of course, you wouldn't So I'm not sure how much it would help. That's the.
That's the interesting.
If you
there
could,
must be some parts of the
if you could sequence a sample of people, I think it might help in some ways. It's quite an interesting question. And it may be possible for people from 50,000 years ago, which would be an interesting time. So I wouldn't rule it out, but I think it would be kind of more interesting for population history reasons than it would be for really sorting out the correct structure of modern diversity.
Just out of curiosity on the X chromosome project, is you have this very large number of reads mapping them down, and I was sort of struck that 30 million reads didn't map back to the X chromosome. What accounts for sort of such a large number? Is that contamination or the reference genome absent in the reference?
No, it's almost all, they do map elsewhere in the genome. It's because when you flow sort the DNA.
So it's contamination in the flow sort.
It's contamination in the flow sort. I mean, now there are some which don't map anywhere in the genome.
Right.
And it's quite interesting where they map. So you can map them to the rest of the database. They map to EBV, because this is an Epstein-Barr virus transformed cell line, and that EBV isn't in the reference genome, so that's one place they map. We know that that should be there. It was in the DNA, and you find it, which is kind of good. They map, some of them map to the Venter sequence, so there's bits of sequence in the Craig Venter version of the genome that are not in the reference genome, which are real. That's an illustration of this extra material argument. Some actually map to chimp, which is kind of interesting. So that probably suggests that there's DNA, again, in this human that is not in the reference or Craig Venter, but is probably human, and the chimp version contains a copy of that. Doesn't mean that this individual was part chimp. It's any more than the rest of us are part chimp. And then there's stuff which doesn't map anywhere, and of course there's just crap. There's sort of stuff which is rubbish. But the stuff which doesn't map anywhere, one can assemble, and people have assembled using, and potentially it's more sequence that isn't in any of the current versions. So that's the answer. Yeah.
Again, sort of a wild question. Would it make sense at all to do sort of sequential sampling? Suppose that you get somebody in some location and you find sort of a new variant
Yep.
or something. And then you might guess that perhaps that variant might be found in people in the same location. That you'd sample in.
Right.
More.
So I think that's kind of interesting. Yes. So I didn't get into the sampling design. I talked about the number trade-off. There's also where in the world you take these people from. And there's a lot of debate about that. But if you ask the question, let's say we find something that looks novel, but we've got low confidence in it. Can we kind of follow up? You could. It's actually hard. You could go back to that person and sequence at high depth in the regions where they have new bits. But at the moment, that's inefficient. It's kind of hard to design something to sequence for a particular person, lots of separate places which are it's 1% of their genome, but the 1% of your choice. There are technologies, people are trying to work on ways to make that more efficient, and it might be that you could design, it might be that it will become, within the next year or two, efficient to do that. You'll sequence each of these people to low coverage, and then where they've got sections that look divergent from everybody else, that are deeply branching, to focus more data collection on those regions. I think it would scare some of the people, the lab people, to have to do that.
Okay, great. Let's thank Richard again.